Welcome to this lecture on the fundamentals of genetics. Today, we are diving into a fascinating topic called epistasis. If you have ever wondered why real-world genetics rarely follows simple rules and why some traits seem to completely vanish, you are in the right place. Let us begin our journey. To understand what epistasis is, let us start with a simple analogy. Imagine you are painting a house and you have two decisions to make. First, you must decide whether to paint the house at all. Second, you must decide what color to paint it. Let us say gene A acts as the master switch that decides if you will buy paint, and gene B decides if the paint is black or brown. If gene A says no paint, it completely does not matter what gene B says. The house stays unpainted. This is exactly how epistasis works. Now that we have an intuitive grasp, let us look at the formal biological definition. In genetics, epistasis is defined as a form of gene interaction, where one gene masks or interferes with the phenotypic expression of another non-allelic gene. The gene that does the masking is called the epistatic gene. Think of it as the boss giving the final overriding order. The gene whose effect is hidden is called the hypostatic gene, acting like the subordinate. Crucially, unlike dominance, which happens between alleles of the same gene, epistasis happens between completely different genes. You might be wondering why this specific interaction is so important to study. Understanding epistasis is absolutely critical because very few traits in the real world are controlled by just one single gene acting alone. Most physical characteristics are the result of complex biochemical pathways. For example, the famous Bombay phenotype in human blood groups is a classic case. A person can genetically have the alleles for type A or type B blood, but they test as type O because a completely different gene prevents the attachment of those antigens. This helps doctors predict complex inheritance. Before we see how epistasis changes the rules, we must establish our mathematical baseline. When Gregor Mendel crossed two individuals that were heterozygous for two different traits, he discovered a very specific mathematical outcome. This is the classic Mendelian dihybrid ratio of 9:3:3:1. The 9 represents offspring expressing both dominant traits. The first 3 represents one dominant and one recessive trait. The second 3 represents the opposite combination. Finally, the 1 represents the double recessive trait. This baseline assumes that the two genes are acting completely independently, without any masking. With our baseline established, let us introduce our first type of interaction, known as recessive epistasis. In this scenario, a gene only exerts its masking effect when it is in the homozygous recessive state. This means the organism must inherit two copies of the recessive allele for the masking to occur. If it has even one dominant allele for that epistatic gene, the masking does not happen, and the second gene is allowed to express itself normally. When this homozygous recessive condition is met, the standard Mendelian ratio shifts dramatically. This specific masking behavior directly alters our expected mathematical outcome, giving us a new formula. The standard ratio modifies into 9:3:4. This happens because the last two categories of the standard Mendelian ratio are combined into a single group of 4. Being homozygous recessive for the epistatic gene completely hides the effect of the second gene. Therefore, any individual with two recessive alleles for the first gene will look exactly the same, regardless of what alleles they possess for the second gene. The distinct categories collapse into each other. Let us see this formula in action with a classic real-world example involving coat color in Labrador retrievers. We are given two genes. Gene B determines the pigment color, where dominant is black and recessive is brown. Gene E determines if the pigment is deposited, where the recessive EE prevents deposition, resulting in a yellow coat. We are asked to find the phenotypic ratio when crossing two double heterozygous black Labradors. We set up the cross and determine the standard genotypes. Because the recessive E allele is epistatic, it will override the pigment gene entirely. Now that we have solved the example, let us mathematically prove exactly how those categories collapsed to give us our final answer. We start with the standard Mendelian output. The 9 puppies with at least one dominant B and one dominant E will be black. The 3 puppies with recessive B and dominant E will be brown. However, look at the remaining 4 puppies. 3 have dominant B but recessive E, and 1 has recessive B and recessive E. Because they all share the homozygous recessive E genotype, no pigment enters the hair. They all become yellow. Having proven how recessive epistasis works, let us explore the alternative scenario, known as dominant epistasis. In this case, the masking gene is so powerful that it only requires a single dominant allele to completely hide the other gene. You do not need 2 copies. Just one dominant allele is enough to shut down the expression of the hypostatic gene. This is a much stronger veto power compared to recessive epistasis. And as you might expect, it alters our baseline mathematical predictions in a completely different way. Because this masking requires only one dominant allele, it changes our baseline ratio differently, resulting in a 12 to 3 to 1 ratio. In this scenario, having just one dominant allele of the epistatic gene is enough to mask the other gene entirely. As a result, the group of 9 and the first group of 3 from the standard Mendelian ratio merge together to form a large group of 12. The remaining 3 and 1 represent individuals who are recessive for the epistatic gene, which finally allows the hypostatic gene to show its true colors. Let us apply this new ratio to a concrete biological example using summer squash. Fruit color here is controlled by 2 genes. Gene W is a dominant epistatic gene that turns the fruit white. The second gene, gene Y, determines color, where dominant is yellow and recessive is green. We cross 2 heterozygous white squashes and want to find the expected phenotypic ratio. We start with the standard dihybrid cross. This time, the presence of even a single dominant W allele will mask the Y gene entirely, making the squash white regardless of the other alleles. Just as before, let us mathematically prove why this specific 12 to 3 to 1 ratio emerges from our cross. We look at the raw genetic output. The 9 offspring with both dominant alleles and the 3 offspring with dominant W but recessive Y all share at least one dominant W allele. Because W is dominant and epistatic, these 2 groups combine to make 12 white squashes. The 3 offspring lacking the dominant W allele can express their dominant Y allele, making them yellow. The single fully recessive offspring becomes green. To truly understand why these ratios change, we must look inside the cell at the biochemical level. Here we see a diagram of a biochemical pathway. It starts with a colorless precursor on the left. Gene A produces an enzyme that converts this into a colorless intermediate. Then gene B produces an enzyme that converts the intermediate into a final colored pigment. If gene A is mutated and recessive, the pathway stops at the very first step. It does not matter if gene B is perfectly functional. There is no intermediate for it to work on. This is the physical reality of epistasis.
We have covered a lot of ground today, so let us summarize our main discoveries. Epistasis is a fascinating phenomenon where genes interact in a hierarchical manner, with one gene masking another. We saw that recessive epistasis modifies the standard Mendelian ratio to 9:3:4, while dominant epistasis modifies it to 12:3:1. Understanding this concept bridges the gap between simple inheritance and the complex, beautiful reality of how traits are actually built in living organisms. You now have a solid foundation in gene interactions.